What's up guys, we are back, and this is the second part of the video of solving questions from the IMAT 2024. In this video we're going to be solving the rest of the questions starting from question 19, and as always I'm going to be highlighting some important points for you to keep in mind in your preparation. Alright guys, so we're going to continue from where we left. We have question 19 here asking about which intracellular structure is composed of microtubules. So this is a very important topic. It shows up a lot in the IMAT, and we're talking about the cytoskeleton. So whenever we're talking about the cytoskeleton, it's important to remember there's three different types of cytoskeleton. Microtubule is one of them. And it's also important to remember that they're made out of proteins. So let's take a look at the options and then we can jump into the details, okay? So we have the letter A here saying that the nucleus is the intracellular structure that is composed out of microtubules. The letter B is saying that it's the Golgi. So guys, if you remember about cell division, you're going to know that the correct answer here is the centriole. It's going to be very important for the cell division, separating the chromatids or the chromosomes. But before we mark letter D and skip this question, let's just refresh your minds about this topic. So as I mentioned, there's three different types of cytoskeleton, okay? The microfilaments, the microtubules, and the intermediate filament. It's important to remember that the microfilaments are going to be the thinner one of these, and also they're going to be very important for the structure itself of the cell, okay? Different shapes that we have from the cells are going to be coordinated by the microfilament. Not only that, but also going to be important for the movement, so the phagocytosis, the pinocytosis, all these movements. And also don't forget, it's going to be important also for cell division, but for the very final step, that is the cleavage, whenever we have to split, okay, these cells into two, it's going to be the microfilament responsible one for doing that. Secondly, we're going to have the microtubules that are important not only for the cell division through the centrioles, but also for all the rearrangement of the substance inside of the cells. And finally, we're going to have the intermediate filament, important for cell division, so one cell with the other, but also for anchoring some organelles inside of the cell. So don't think that the organelles are going to be floating like randomly inside of the cell. They're all placed where they should be. So yeah, these are the three names that you have to remember along with the functions. By doing that, I'm sure you're going to be good to answer any questions related to this topic. So once again, the correct option here for question 19 is letter D. Alright, so question 20, mitochondria have. So I think this is the third or fourth question just talking about mitochondria, so you can see how important this topic is for the IMAT. So let's remember the structure of the mitochondria. This is a very interesting organelle that shares some past with the bacteria. If you remember about the theory of the endosymbiosis, you might remember that it was once a bacteria that entered our cells, and through a symbiosis process, it remained there. So this has its own membrane, just like the bacteria, and also has an outer membrane that supposedly is the one that it received when it first entered our cells through phagocytosis. So having this picture in mind, it already allows you to eliminate a couple options here. So letter B, for example, an outer membrane consisting of a phospholipid monolayer. Now we know that a membrane cell has a double layer, right? As a phospholipid bilayer. So it's not the case here. Letter D, an outer membrane and an intermediate membrane and a very selective inner membrane. So it's telling us that there's three membranes. That's not true. Where the intermediate membrane is coming from. So that's not what we're looking for. Letter E, it's a very selective membrane in which no proteins are present. If you're talking about membrane cells, we're talking about phospholipids, we're talking about proteins, we're talking about cholesterol. So all these has to be present. So this leaves us with letter A, only a very selective outer membrane, and letter C, an outer membrane and a very selective inner membrane. So they're kind of the opposite of each other. But if you may, let's remember that the inside of mitochondria, there's so many things going on there. So we have oxidative ions there. We have the cytochrome C, for example, that if it's released of the mitochondria, it kind of stimulates a reaction of apoptosis. And that's not interesting. So the inner membrane has to be very selective to decide what comes in and what goes out. So letter C is the perfect definition of it. Then moving on, question 21 is asking what is an anticodon. So mainly this question is talking about the central dogma of biology. No, it sounds a little bit prophetic, but basically we're talking about a very well established topic in biology, that is protein synthesis. Okay, we're talking about translation, we're talking about transcription, and specifically anticodon, we're talking about the protein of amino acid, okay, building polypeptides. So with this in mind, let's take a look at the options. Let's see what does it fit in this description. So letter A, it's a part of the DNA that codes for a specific amino acid. So DNA, protein synthesis happen close to the DNA, not necessarily, there's a step in between, right? The letter A is not what we're looking for. Letter B, the sequence of three nucleotides found on a tRNA corresponding to a codon on the mRNA, so the messaging RNA. Perfect. This is the best and most complete option. If you don't remember this, let's take a look of this little scheme here. So basically, while producing proteins, we're gonna have the DNA as being the main template for this, okay, and we're gonna have a process that we're gonna call transcription. So we're transcripting section of the DNA. Remember that DNA has genes, and this section is gonna be transcripted into something that we're gonna call a mRNA, so a messenger RNA. After that, this messenger RNA is going to leave the nucleus through this porous that we can find in the karyotype, and it's going to try to find a ribosome. And this is going to be the main character to produce the protein. Once it finds it, it's going to be read by a tRNA, a transfer RNA. Now, this is a very interesting because a tRNA is the main character of the translation translation. The ribosome is going to be important for accommodating everyone, but a tRNA is going to be the one that is going to carry the amino acid. And the other extreme of the tRNA, we're going to have the letter code that is going to pair perfectly into some of the sections here of the RNA. So think about this in a writing perspective, okay? So for example, transcription is something that a lot of medical students do while studying for a hard subject. For example, someone is responsible for recording a class. I know in many places that it's not allowed, but there's so many important details in that class that you can't split your attention between writing down and paying attention to the teacher. So a lot of students just recorded classes and later on in their houses they just transcript. So it's basically reading and transcripting word by word, letter by letter, whatever the teacher is saying. So once that is done, you have basically a piece of paper that is identical as what the teacher said in class. Now imagine you wrote that all in English and you want to translate this into your mother language. So basically that is the translation. You're getting that piece of paper, you know, that have those words, and you're changing completely the letters to form something that makes sense in your own language using your own grammatical rules. So you're going to change the subject. Instead of using nucleotides, that was the language used in the transcription, you're going to use amino acid. Alright, so moving on, question 22, what are ribosomes made of? Very straightforward, and if you may, let's just remember ribosomes, important organelle for protein synthesis. Where are they made? In the nucleus. So if you remember that, you might think that, okay, if they come from the nucleus, they might be made out of DNA. Wrong, okay? They're made out of RNA. That's the most important thing to remember. And what? And proteins, all right? Yeah, ironically, they make proteins and they're made out of protein. So letter A is what we're looking for. They're not made out of DNA, so none of the options here will be correct if you remember that. So just a quick reminder, okay, we have the nucleus here, very important, different roles. But most importantly, let's just remember that we have a very dense area inside of the nucleus that is going to be the nucleolus. You can even see here in microscope, okay, electronic microscope. Other than that, because we're talking about the nucleus, let's just remember that we're going to have the DNA mixed with histones that are going to be important proteins that will organize in some way the genetic material. And whenever we don't have a situation of cell division, this DNA is going to be unpacked in a way that we're going to call it chromatin. They're not going to be chromosomes. We're only going to have chromosomes when we have cell division. Moving on, question 23. The cell membrane consists of, so once again, let's just picture the cell membrane. We're going to have a double layer of phospholipid. And the outer side, we're going to have the phosphate, and the inner side, we're going to have the fat chain. Other than that, we're going to have proteins, we're going to have cholesterol, and also glycocalyx, okay, or glycoproteins. Let's check the options here. So it's a double layer of triglycerides and cholesterol. No. A double phospholipid layer with hydrophobic tails facing inwards, and the presence of an integral and peripheral proteins. Okay, this looks correct. Let's just refresh our minds what all this means. So as I was saying, we're going to have the phospholipid here that is going to have a hydrophobic tail. That is basically the fat chain. Other what I was mentioning, an integral protein. What is that? It's basically a protein that crosses through the two layers of the membrane cell, okay? So we're going to have channels like this that allows ions and other substance to go in and out of the cell. We have this transmembrane protein. We're also going to have a peripheral proteins, okay? They're basically proteins that doesn't cross the two layers of the membrane. They're going to stick to one of the two sides of this membrane. So what is the importance of these proteins? Proteins are not going to allow substance to come in and out of the cell, obviously, but they're going to be very important for cell signalization or cell anchoring, for example. Finally, we're going to have other two important components of the membrane cell. There's going to be the carbohydrates forming the glycocalyx or the glycoproteins, also important for the cell signalization, and the cholesterol, very important for controlling the fluidity of the cell in situations of heat or cold. All right, so nearly there. Question 24. In protein synthesis, what is translation? So once again, we're talking about the central dog in biology, okay? Who are the main character of the story? We're going to have the mRNA that is going to be read. Where? In the ribosomes, okay? They're going to accommodate all these people. And the tRNA or transfer RNA that is going to carry the amino acid that is going to build the polypeptide, and also that will pair exactly where it should in the mRNA through the codon and the anticodon. With that in mind, let's take a look at the options here. Letter A, it's the process in which DNA is read and the corresponding mRNA produced. So is DNA being read? Not. We're talking about translation, not transcription. Letter B, it's the process of pairing between DNA codon and tRNA anticodon. DNA codon, does that even make sense? Not at all. C, it is the process by which mRNA is read and converted into a specific sequence of amino acids. Perfect. That is the perfect definition. We don't have to go any further. Letter C is the correct option for 24. All right, so question 25. What are the principal components of the cytoskeleton? So cytoskeleton, once again, well, that's going to be easy. We already have fresh in mind. What are the three different types of cytoskeleton that we have? First of all, we're going to have the actin filaments or the microfilaments. Other than that, we're going to have the microtubules. And finally, what? The intermediate filaments. So letter D is the only one that points out this, okay? The other ones will be distractor. We have letter E saying actin, myosin, and dynein. All right, these are going to be very important for what? For cell contraction and protein movement. But they're not properly types of cytoskeleton, okay? Even knowing that they're related to the cytoskeleton in some way. And finally, question 26 is asking the term allele defined. So what is an allele? So we're entering the genetic questions that we had that year, and this is a very fundamental topic of genetic, okay? So what is an allele? You must remember that when we're talking about allele, we're talking about different types of what? Of genes, okay? And what is a gene? Gene is going to be a section of the DNA that is going to be responsible for producing proteins. So they're going to be very important not only for creating the features of an individual, but also responsible for disease and stuff like this. So in this case, we're gonna have a lot of distractors here, but the correct option is letter E, one of the several alternative forms of a gene. Questions like this that are very fundamental, you might find an option here that is correct in some way, okay, but does not define or does not answer the question itself. So that's it for this video, guys. I hope you guys enjoyed, and if you did, don't forget to like and subscribe to the channel if you're new, and if you have any questions, just fire away and I'll get back to you as soon as possible. I see you guys in the next one.